Welcome to this edition's Spotlight Science, where we put science stories in the spotlight. Our today's guest is Professor Yinon Ben Neria from Hebrew University in Jerusalem, and he and I will be talking about his recently published Nature paper, where he and his team discovered that the tumor suppressor protein, p53, can actually have both tumor suppressive and oncogenic effects, but it depends on where in the gut the tumor suppressor protein is located and, surprisingly, on the gut microbiome. Enjoy the dialogue. Thank you very much and welcome. Today we're going to talk about the paper you and your group did, where basically you saw that the tumor suppressor protein p53, when you do a mutation on it, can be actually both oncogenic and tumor suppressive, but depends on a few factors, and the microbiome is probably the most important factor. So first of all, I would like to ask you about the whole hypothesis behind p53. So when you made the hypothesis, did you specifically aim at p53? And was the initial hypothesis that p53 mutation can cause both oncogenic and tumor suppressive actions, or maybe something else?
Yeah, well, we didn't guess that when we started. So p53 mutations are very special. p53 itself is a tumor suppressor protein, gene and making a tumor suppressor protein. But if it is mutated, then you lose the tumor suppressor function of the protein. But unlike other tumor suppressors that are, most of them are tumor suppressor, we know they are tumor suppressor because when they are deleted, it supports tumorigenesis. Unlike most others, or all others maybe, some p53 mutation, it's actually maybe a majority of the p53 mutation, what we call missense mutations, which means the protein is made, they behave as oncogenes. So not only that you lose the tumor suppressor function, you gain an oncogenic function. And this is very unusual for tumor suppressors, as I said. I mean, the other, like APC, for example, is another tumor suppressor which is relevant to us in the gut. Basically, you lose it in cancer, that in 90% of colorectal cancer, it's lost. But so the mutation either inactivated, or you don't have the protein made for some reason, or it's shorter and so on, but never you get a gain of function APC mutation, which means it never turns into an oncogene. So 50 gain of functions are unusual, and we don't clearly understand this phenomenon very well.
Mm-hmm. So you were surprised by the results of that your group made?
No, we knew that, and there is a lot of interest in trying to understand mechanistically what is gain of function and how does it work. We do animal studies on signaling and mutations. Cancer mutations are important because, first of all, we want to understand the disease development, cancer, and also the mutations are the basis for diagnosis and for therapy. Most of the biological therapies against cancer are based on mutated proteins like EGFR, kinds of different molecules that are targeted. So we said, let's try and express the mutant, this gain of function, this very peculiar gain of function mutant of p53 in the gut, in the mouse gut, and see what it does. And we had very simple expectation. We thought we knew before that if you knock out, if you delete the p53 tumor suppressor genes, together with deleting CK1 alpha, which is an enzyme that we like, you get cancer, you get intestinal cancer. So we said, if we now express the gain of function mutation of p53 together with the CK1 deletion, we should at least see cancer all over the gut like we saw with knockout of p53. So that was the expectation, and it was fulfilled only partially. And the big surprise was that when we looked at the jejunum, at the proximal gut, the duodenum and jejunum, we didn't see any cancer. Even so, CK1 alpha by itself doesn't induce, when you delete it, you don't get cancer, but mild dysplasia, which is, one phenotype of cancerous cells. And this was also disappeared totally when you actually express the mutant. And just imagine, it's an oncogenic protein, very strong oncogenic protein. And all of a sudden, you don't see anything in the proximal gut.
Now, yeah, that was surprising to me, actually.
Very surprising. So that was the first puzzle. And then we looked at the other part, and we saw what's expected. So in the distal gut, which is mainly the colon, you see cancer, which actually you saw the gain of function. You saw that it's worse than deleting the 53 protein. And then we became interested in the first phenomenon. Why is it, how come it's a tumor suppressor? All of a sudden, an oncogenic mutation turns into a tumor suppressor. So the clue was the Wnt pathway. So the Wnt pathway is the driving pathway of colorectal cancer and some other cancers as well, liver, and so on. Gate opening the mutation in colorectal cancer is the APC mutation. That's a gatekeeper. So if you lose it by some mutation, then you start to have benign tumors in the
mm-hmm.
So we also use a model based on APC mutation. And so this is called APC mean. It's a classical model, almost study that works on intestinal cancer, uses this model. And all of a sudden, we saw something which was similar to what we find in the other model. So in mice, there is that relates to one of the questions you asked. The APC mutation causes benign tumors, actually only in the jejunum and not in the colon. In humans, the APC mutation, which, as I said, it's 90% of the colorectal tumors are initiating probably with the APC mutation. The tumors are found in the colon. And usually it starts with benign tumors in the colon. But in mice, for some reason that we don't understand, we find the tumors mostly in the jejunum, in the proximal gut.
Which is represented here, yeah.
Right. Nobody understands this. I just want to mention something about the model. The model harbors heterozygous germline mutation in APC.
Mm-hmm.
So initially you don't have any cancer, any tumors in the mice, but when they grow about 4 or 5 months old, they undergo a process which is called loss of heterozygosity. The other allele of APC is lost, and then they start to have benign tumors. And that's what you see here in the left panel, right? You see in the jejunum, for some reason, many tumors which are marked by arrows here.
Mm-hmm.
Why is that different from the humans? We don't understand. Maybe the rate of loss of heterozygosity is different between the jejunum and the colon in humans and mice. But nobody understands. Anyway, that's the model we start with. And now we introduce the 53 mutation, the gain of function.
Which is here? Mm-hmm.
Exactly. And this is, I think, the second or first, one of the two most common 53 gain of function, 53 mutations in humans. It's called the hotspot mutation, the R172. So the expectation was that if we introduce this oncogenic mutation on top of APC, you'll see many more tumors, both in the jejunum and maybe in the colon as well.
Mm-hmm.
But all of a sudden, as you see here, you don't see any tumors in the jejunum and the proximal gut, and you start to see many tumors in the colon. So it reversed the pictures of just APC by itself.
It's almost paradoxical, isn't it?
Yeah, that's we already had some information from the other model, so we weren't surprised this time.
Mm-hmm.
Uh, we saw that in the jejunum, also in the other model, the introduction of the p53 mutation totally eliminated any tumorigenesis in the proximal gut. So
Mm-hmm.
that was actually a nice confirmation for the other model.
I think it's really surprising, that you see a lot of tumors in the APC mutation and wild type p53, and then when you make a p53 mutation, all of a sudden the tumors kind of almost disappear,
Right.
basically. So that's
Right.
really, really surprising, at least to me.
Right. Now, the question is whether we have a clue to the mechanism of
Exactly.
tumor
Mm-hmm.
suppressor. And then we went to the Wnt pathway,
Mm-hmm.
which is, as I mentioned, is a driving force of tumorigenesis in the gut.
Mm-hmm.
And we saw that basically the p53 gain function mutation wiped out the Wnt pathway gene expression in the gut.
Yeah.
Which then it intervenes in the with the Wnt pathway signaling, and consequently you don't have any Wnt gene expression, which is the basis of the tumorigenesis.
And speaking of the Wnt pathway, I can also show another figure from your paper, figure 5 here, and we can maybe focus on figure 5D, and you can maybe explain just a tiny bit more about this mutation CK1 alpha with p53 mutant and what we basically see here.
Right. So yeah, this one, so the light blue spots are the most interesting ones.
Mm-hmm.
And so these are mice which express the both CK1, deleted CK1 alpha together with the mutant p53. And here you see no binding of the transcription factor TCF4 to Wnt promoters. So signaling in the Wnt pathway is dependent on two factors. One of them is the TCF4, which is the DNA binding factor, and the other is called beta-catenin. Beta-catenin binds to TCF4, and together they induce gene expression, Wnt gene expression. And all these are Wnt genes like AXIN2, CD44, cyclin D1, and SOX9 are targets of the Wnt pathways, which you should see, and they should be active in the gut. And here you see that basically p53 mutant expression totally shut them down. So the TCF cannot bind it to the chromatin, and you don't have any Wnt gene expression.
Yeah, and you don't basically have Wnt activation and basically reduced level of activity, meaning reduced number of cancer tumors.
Right. So without hyperactivation of the Wnt, you don't see intestinal cancer, and that's what we see in the jejunum, right? I mean, this is specific for the jejunum.
Yeah. Did you maybe look at other sites of the gastrointestinal system apart from the jejunum?
If you look at the mirror image in the ileum, for example, or the colon, you see very nice TCF binding to this promoter. So this is specific for the proximal gut. We're only in the proximal gut. Right? So that explains why in the proximal gut you don't have tumors. No Wnt expression, no tumors.
And then you basically chose one of the metabolites in the gut, which is called gallic acid, and then you saw the TCF4 enrichment is much higher in these mice than in the mice without treatment with gallic acid, and that probably
So the idea was that bacteria may be the clue to understand the difference between the different parts of the gut. The bacteria are much more abundant in the distal gut, in the colon, than of the jejunum and duodenum, for example. And we confirmed that. We treated our mice first with antibiotics, the ones that have mutant p53. And as I told you, in those mice, only the jejunum was free of tumors. But when you treat the mouse with antibiotics, and you clear the bacteria from the distal gut, from the colon, all of a sudden you see no tumors anywhere. So, I mean, basically, you got rid of the tumors in the entire gut. Let's see what's in the bacteria is responsible for that dramatic effect. So, fortunately enough, there is now a very nice methodology which was initially introduced by Hans Clevers, and for making mini gut, mini organs. These pieces of gut, actually you can start from even a single cell, single stem cell in the gut, on which you grow organoids, and they in a way mimic the gut structure. They have, for example, Vilus and Crypts equivalents. So, and you can say whether they are differentiated, meaning having these structures, or looking more like spheres, non-differentiated, with no structures at all. When you take organoids, for example, from gut tumors, from the APC mice, for example, they never differentiate. They are round, they're called spheroids. We took differentiated organoids from our mice, said, let's try and treat them with various metabolites from the bacteria and see whether any of the metabolites can turn the differentiated ones, which mean the normal ones, to spheroids, to like tumor-like ones, which no structures at all. So that was the basis of the screen.
And you also, you tested short chain fatty acids. I've seen in the paper, right? And you saw no effect.
Right, no effect at all, like butyric acid, and also other metabolites as well. And eventually we focused on one group of metabolites, which has the capacity to round up these organoids. And then we narrow down on one, which is gallic acid, which doesn't only round up the organoids, but also induces massive proliferation and Wnt expression in those little structures. First of all, actually, we looked into the gut and to see whether gallic acid is produced in parts of the intestinal gut, of the gut, and we found indeed the gallic acid only in the distal gut, which means in the colon, ileum. Where the bacteria are, and that actually told us that probably the bacteria are responsible for making the gallic acid in the mouse gut. In the proximal gut, there are hardly any bacteria, so indeed we didn't find any gallic acid. And we said, okay, let's treat now, let's feed now the mice with gallic acid and see what happens to mutant p53 with CK1 alpha. And when we did it, and that's part of these figures, all of a sudden, mutant p53 loses its tumor suppressor function and reverts to be oncogenic. Which now you see that all the Wnt promoters are becoming active and sometimes hyperactive. Right? And that allows regaining of the Wnt signaling pathway and Wnt hyperactivation, and you see cancer, you see tumors.
Yeah, we can see basically in the picture here, on the left, the difference between the group that's not treated with anything and the group with gallic acid, that the group with gallic acid in the proximal gastrointestinal tract, meaning the duodenum and jejunum, have significantly more tumors than the group not treated with gallic acid. Basically, and did you maybe did you maybe take a look at if these tumors were benign, malignant, or like a mix of everything?
So in this model, it's you don't reach the malignant phase because the mice, they cannot tolerate it. They have so many tumors and they die from anemia because they bleed. Tumors are bleeding. So it's not a great model. In the other model of the CK1, actually you see massive carcinogenesis. I mean, what you call high-grade dysplasia all over. So you can confirmed that it's actually malignant transformation of the entire gut. That's,
and have you maybe so you told me that you selected out of you did like a screening for metabolites in the gut and you saw that gallic acid had a strong effect, but did you maybe try combining the metabolites, maybe short chain fatty acids plus gallic acid, or did you just selectively put one by one?
So we focused on gallic acid, but that surely doesn't tell you that there are other metabolites, bacterial metabolites, can do the same thing. We showed by clearing the gut from bacteria by antibiotic treatment, massive antibiotic treatment, that gallic acid is sufficient to transform. But we didn't prove that this is the only one that does the job. And this is important because for gallic acid, we actually are familiar with the biosynthetic pathway that is responsible for gallic acid production. This synthetic pathway is only found in bacteria, plants, and fungi, not in animal cells. So, mice and humans don't have this biosynthetic pathway. So we can think of even measures to try to inhibit the synthesis of gallic acid. But whether it will help, we are not sure, because if there are other alternatives, there are other methods that can do the same job, then we have to look for them and target them specifically as well.
So would your future steps maybe in this research be to look for other metabolites or to focus on something else?
Right. So we have both. We try to target the gallic acid biosynthetic pathway on one hand, but try to look for both specific bacteria that are associated with this phenomenon of mutant p53 induced transformation, and then see whether there would be other metabolites, and we have people helping us.
Well, how long would you say it took from hypothesis forming till publishing the paper? How long was this research like approximately? Yeah, I was always wondering that.
Well, first of all, when mice are involved, it always takes very long. So you have to generate the models. This is by breeding mice, different mutations and so on. And then you have to wait until tumors come up. They're not born with the tumors. It takes time and so on. So I think the whole process took something like 5 years.
So just maybe shifting from the paper to your scientific experience in the past, I've seen that you've also been a researcher in leukemia field. And together with your colleague, you developed inhibitors for the BCR-ABL protein, which eventually led to the discovery of Gleevec, imatinib for chronic myeloid leukemia. So are you still interested in leukemia? Are you still in that research, or maybe not?
We still are, from a different angle. So we, working on the Wnt pathway, we characterize critical proteins in the Wnt pathway, and one of them is CK1 alpha, for example. So we, like 10 years ago, we started to develop inhibitors of CK1 alpha, because we found that if you block CK1 alpha, you actually induce p53. And that's the normal wild-type p53 is, of course, a different story, is something good to induce. So we developed inhibitors, and we were fortunate to work on a leukemia model, which was the easiest model to see that those inhibitors are of value. And some of these inhibitors are, so these are CK1 alpha blocking small molecule inhibitors. And they have some other interesting activities. They also block transcriptional kinases. And together, this combination is very useful to kill leukemia cells. But now we are not talking about CML, chronic myelogenous leukemia. We're talking about acute leukemia, acute myeloid leukemia. And we were fortunate, and the lead compound is now in phase 1 clinical trial.
Regarding the signaling pathway, since you're focusing on the signaling pathway, what would be your ultimate goal there? Maybe to discover a drug that can maybe modulate and suppress cancer, or maybe something else?
We actually focus, as I said, we focus on some signaling pathway that we are familiar with. And yeah, I wish we had thoughts to find something, since Wnt is so important in many tumors, we thought of trying to tackle this pathway and find some inhibitors. Many people are actually interested in that as well, and so far we don't have very good inhibitors, very good drugs to tackle Wnt and so on. We are still active, but our main interest is still basic research, like find out how cancer develops. I mean, seeing something like a mutation that changes its face, like Dr.
Yeah.
Jekyll Hyde and so on. I think that gives us still more satisfaction.
What would you say for maybe younger scientists who are just joining the field of science, just starting into science? What would be like the best advice you could give them? Maybe the advice you would give yourself when you started
Right.
research.
So many students come to their mentors, and the mentors have a proposal, something to look for, and they start working on something, and then they observe something different or unexpected, and they come to their mentor and say, "Hey, I noticed something very strange." And he said, "Don't worry about that. Just go and continue with the line that we actually decided to work on." My suggestion is to stick. If you find an interesting observation, don't let it off. Whether your mentor is happy about it or not, you have to find a way to do that and try to understand it. And because discoveries are made this way, not by looking for what you are naturally expecting, but for something that you don't expect. And I think curiosity, if curiosity gets you to something unexpected, that's even more encouraging. I mean, it's something more rewarding than anything else.
Yeah, I totally agree, and I understand what you're saying when some mentor tells you maybe to do something and then you find something else and then no, stick to that. I understand, but that's great advice, really
That's
great.
very common. I've noticed it myself, but
Do
it's
you tell
strong.
that to your students?
Yeah, no, to my students, I actually tell them, I mean, if they show me something unexpected, I never ignore it. I usually try to understand what's going on. And that's I think, is, as I said, very most rewarding to follow up the unexpected.
Yeah, follow the unexpected. And Professor, it was a pleasure talking to you. Thank you very much for joining us from Hebrew University in Jerusalem on this research dialogue in our Spotlight Science.